together to produce energy in the form of ATP. So, but before we start that, can one person loudly explain the mechanism of action of the G-sub protein signal? One volunteer, please. Yes. Can you be a little bit louder, please? Sorry. Excellent. Did you guys hear what he said? Perfect. No? Did you want me to repeat it? So that was very, very spot on. So what happens is you have an inhibitory ligand binds to an inhibitory G-coupled protein here. That's going to cause the alpha subunit and the beta gamma dimer to separate. That separation is going to cause this alpha subunit and the associated GDP to be converted to GTP. GTP, this alpha subunit with the GTP coupled is going to bind with the adenylate cyclase enzyme, inhibiting it, thus inhibiting the conversion of ATP to cyclic AMP and inhibiting this entire pathway as shown here. Any questions about that? Okay, so can anybody, one person, tell me what are the inhibitory G-coupled protein receptors that we talk about? MAD2, love it, yes. Muscarinic 2, and the ligand or agonist is acetylcholine, alpha adrenergic 2, and the ligand is epinephrine, norepinephrine, and dopamine 2, and the agonist is dopamine. All right? Any questions about that? All right. Well, yesterday we talked about different phospholipids and how they're involved in making up the cell membrane. One of the phospholipids, cardiolipin, one of its roles or function was to help decrease blood clot formation. And how it does it is a complicated and involved process, not fully studied, but what happens is there's a condition called APS, which is an antiphospholipid syndrome, where a patient has an autoimmune issue, and the issue creates antibodies against cardiolipin, lupus anticoagulant, and another glycoprotein. And so that would cause the recurrent blood clots and the spontaneous abortions or spontaneous stillbirth that I mentioned yesterday. And so what cardiolipins do is that when they're functioning properly, they not only help to maintain the mitochondria and cell health in general, but they also help decrease the risk of blood clot formation. And so as clinicians, when you guys become PAs and you suspect a patient has the APS syndrome, you would order the clinical lab tests that are tests for the different antibodies against the items shown at the bottom of the screen. Any questions about that? Somebody tell me the mechanism of action of calcium channel blockers. One person. Anybody? Please? Yes.
Sure, I'll try. So essentially, they block
they block the calcium channels, and then what happens?
So then it brings down your rate and your blood pressure.
Perfect. Absolutely perfect. So calcium channel blockers, they decrease blood pressure. How they do it is they decrease calcium from entering muscles, and that's going to eventually lead to increased diameter of blood vessels to decrease your blood pressure. Okay, does that make sense? Everybody got that? Thank you very much. Great job. Amlodipine, of course, we talked about the D-pin drugs are an example of calcium channel blockers. One of you scholars asked yesterday, needs to be taken during pregnancy. The answer is yes. Nifedipine can be taken. It's preferred because it's been the most studied during pregnancy. However, it is recommended that if it is prescribed to a patient, it should be done during the second and third trimester, because the first trimester is just risky in general for giving drugs like this. They're not 100% sure if it's teratogenic. They don't think it is based on their studies so far, but it can be used during pregnancy. All right, can somebody tell me what stage of signaling this is? I know somebody knows in the back. Yes. Close. Transduction. Yes. Yes. Yep. Hey. You volunteered. I appreciate you. Yeah, this is transduction. All right. Then got another question about histamine. We know that histamine 2 is also blocked, and that is used during when a person has acid reflux. Histamine 2 receptors are G sub S coupled. We know this coupling causes the stimulation of cyclic AMP in that cascade that we talked about. But these histamine 2 receptors are found in the stomach primarily, and they cause the increase of gastric acid production or secretion from parietal cells. So, of course, we would give histamine blockers because we don't want that acid to be overproduced. Now, histamine 1 receptors are G sub Q coupled, and these receptors are essentially pro-inflammatory. So what they're going to be doing is causing sensory nerve stimulation that's going to cause us to itch. In the GI tract, they're going to cause muscle contractility, which can lead to diarrhea. And in our tissues, it's going to cause increased permeability of fluids, and that can lead to edema and hives. So pro-inflammatory responses. Remember the G sub Q pathway. G sub Q is different than G sub I and G sub S. It's a unique pathway, and it causes the activation of phospholipase C. PIC2, phosphoinositol bisphosphate, is cleaved into DAG, diacylglycerol. DAG hangs out in the cell membrane. IP3 leaves the cell membrane, and then it causes endoplasmic reticulum release of calcium, and that causes the cascade of effects that lead to the physiological effects shown here at the bottom. All right. Any questions about this? All right. This next question that was asked shows that my old brain, even I don't remember everything, and I've seen this stuff, looked at this stuff for years. So it was asked yesterday, are PE and PC sphingophospholipids? They are not. They are glycerophospholipids, and they're actually ones we talked about in the last block, and I should have remembered that. I didn't, but I'm getting old, so I'll use that as an excuse for everything. And so they are phosphoinositylcholine is lecithin, and phosphatidylethanolamine is cephalin. Any questions? Right. Now we're going to move into the topic of today, the TCA and electron transport chain system cycles. So look at that. We're almost halfway done with this metabolic pathway table. So in the TCA cycle shown here at the bottom, this cycle is the central hub for biochemical metabolic pathways. This is where we see that the macronutrients that we discussed in Block 1, they all merge here with the ultimate goal of going through this pathway to produce reduced coenzymes that then travel to the electron transport chain to give us energy from ATP. Here is a schematic of the TCA cycle. We're going to go through it in a little bit more detail, but you guys don't need to know the structures. You don't need to know every single output. We're going to talk about what you need to know. So there are some slides that might be hidden that give the details of the pathway. You don't need to, unless you're interested, none of that information is going to be on the test. It's just the specific details of every single step in the pathway. And so in general, the TCA cycle occurs under aerobic conditions. And so what happens is during the first step, that acetyl-CoA combines with oxaloacetate, which is the final molecular product of the pathway, to produce citrate. That's how the cycle gets its name. Citric acid cycle. Citric acid is a tricarboxylic acid, and that is how it got its name. Also, the scientists that discovered it, Krebs, so it's called all three of those. So, what happens in this pathway is we gain reduced NADH, FADH2, and GTP. Those are the main products of this pathway. That GTP is converted to ATP in a different part of the cell. And so you don't need to know the different types of reactions in terms of hydrolysis, hydration, but you already know, because since we're gaining reduced NAD and FAD, you already know there's going to be oxidation and reduction reactions, right? Because the substrates are going to be oxidized in order to give us the reduced coenzymes that are going to go on to produce the ATP. So in this diagram, it's showing how all of our metabolism or catabolism, the breakdown of molecules, converge at the TCA cycle. And so there are three different stages. What happens is the first stage, we consume macronutrients, proteins, polysaccharides, and lipids. Those first are digested into smaller molecules. Those small molecules enter our bodies and are then broken down further if need be and converted to acetyl-CoA. This conversion to acetyl-CoA occurs in the mitochondria. So this is where all of these macronutrients merge at acetyl-CoA, and then, as I mentioned, acetyl-CoA then produces the FAD, H, and NADH in order for us to get energy, and this occurs, that final step occurs in the electron transport chain. Any questions so far? So, as I mentioned, the TCA cycle occurs when oxygen is present. So what needs to happen first is that pyruvate molecule that we attain from the final step in glycolysis is oxidized to acetyl-CoA. And what happens is, when it's oxidized by pyruvate dehydrogenase, it combines with the CoA moiety. Remember that HS thiol group is very reactive, shown here in green. And that pyruvate dehydrogenase enzyme transfers its electrons and hydrogens to the NAD to give us the reduced NADH, which also will travel to the electron transport chain to produce ATP. So this is just a schematic of acetyl-CoA synthesis. So glycolysis occurs in the cytosol. So a carrier needs to transport that pyruvate into the mitochondria to carry out that conversion to acetyl. So the final products of TCA are carbon dioxide. That's not really important. The important final products are 3 NADH, 1 FADH2, and 1 GTP. And this would be with 1 molecule of acetyl-CoA. So if we start from glycolysis, remember glycolysis produces 2 pyruvate molecules, thus 2 acetyl-CoA molecules. So you would just double that number. So starting from glycolysis, we would achieve 6 NADH, 2 FADH2, and 2 ATP molecules. Any questions? And this is what I want you guys to know about the TCA cycle. So the first step, acetyl-CoA combines with oxaloacetate to produce citrate. Citrate is converted to isocitrate. Isocitrate is converted to alpha-ketoglutarate. This reaction is important because it's the rate-limiting reaction catalyzed by isocitrate dehydrogenase, and this is where we gain our first NADH molecule. So rate-limiting enzymes are like the bottleneck enzymes. They control how fast the reaction rate goes, and they're also typically regulated by the product. So if you have too much ATP, for example, isocitrate dehydrogenase is going to slow down. So, the reaction to occur so fast. So that alpha-ketoglutarate is converted to succinyl-CoA. We're going to talk about succinyl-CoA. It is very important in the production of heme. We're going to talk about that in a future block. Here we gain our second NADH molecule. Succinyl-CoA is converted to succinate, and this is where we get the GTP molecule that is converted to ATP in a different part of the cell. And then that succinate is converted to fumarate. This reaction step produces the one and only FADH2, and then fumarate is converted to malate. Malate is converted to the final molecular substrate of this pathway, which is oxaloacetate, and this is where we gain our third and final NADH enzyme. Any questions? Yes.
We're not concerned with what turns citrate into isocitrate.
No. Any other? All right. So, where did we see oxaloacetate? Does that sound familiar? Say it again. Yes, where else? We did see it. We did. Where else have we seen it so far? Hey, yes, absolutely. Gluconeogenesis. Yep, we saw that. So this shows you how all of these intermediates and products kind of floating around in the cell, and depending on what the body needs, it's going to use it as needed. And so, yes, great job. So oxaloacetate is the first step in gluconeogenesis. All right. Check on them. So, in one turn of the TCA cycle, how many carbon dioxides do we gain? 2. That's not. I'm glad you guys know it, but that's not the main products. But I'm glad you guys know it. What about NADH? In one turn. One turn. 3. Right. What about FADH2? 1. And GTP? Perfect. Absolutely perfect. Great job. Any questions about the TCA cycle? Now we're moving on to powerhouse, the energy producer, and that is the electron transport chain. So here's the schematic of the electron transport chain. As I've said before, the reduced coenzymes travel to the electron transport chain and they donate their electrons and hydrogen ions to this chain in order to produce ATP. There are 5 complexes in the electron transport chain. 1, 2, 3, 4, 5. The 5th one is ATP synthase. That is where and how ATP is synthesized in this pathway. And so let's go over the pathway in detail by each of the different complexes. So we know that most of the NADH, most of the NADH and the FADH2 from the citric acid cycle, they're already in the mitochondria. So they don't need to be transported. But there are other places that we get NADH that are used to produce ATP. Can you guys tell me some other places we get NADH and the pathways that we discussed? Is one person saying it? Glycolysis, absolutely. Where else? I didn't hear. The PPP. Do we get NADPH? Close. But then that's very important as well. What about what we just talked about with pyruvate oxidation? So when pyruvate is converted to acetyl-CoA, that actually, the process starts in the cytosol, but then it moves to the mitochondria. So what happens is, particularly for glycolysis, that NADH that is produced in glycolysis needs to be able to the electrons and hydrogen ions to be able to be transferred into the mitochondria. And the NAD molecule cannot enter the mitochondria from the cytosol. So what happens is there's a malate aspartate shuttle, and that will transfer electrons from cytosolic NADH into the mitochondria, and it does that by transferring the electrons from oxaloacetate to malate. Malate can then enter the mitochondria from the cytosol, and malate will transfer its electrons to an NAD to regenerate oxaloacetate. You guys don't need to know this step, that's why there's no diagram. It's just showing you that there's a way that the electrons and hydrogens from the NAD attained in glycolysis enter into the electron transport chain. So that's one way, and that's the malate aspartate shuttle. And so again, the goal of the electron transport chain is to produce ATP. There are 5 complexes, numbered 1, 2, 3, 4, 5, and there are 2 electron carriers, ubiquinone, or also called OQ, and cytochrome C, and these electron carriers will transport electrons from the complexes to its final acceptor, which is the oxygen associated with complex 4. So in the first complex, NAD will transfer its hydrogens and electrons to complex 1. I got asked a great question as to why NADH only works at complex 1. That's because it has a specific binding affinity to the proteins associated with complex 1. So that's why NADH specifically works at complex 1. Same thing for FAD. FAD has binding specificity for complex 2. So what happens in complex 1? NADH donates its hydrogen ions and electrons. The hydrogen ions, once the electrons are donated, that's going to cause a conformational change. The hydrogen protons are going to be pumped into the inner membrane space shown here. And then that NADH is oxidized to NAD. NAD is then able to pick up more electrons and hydrogen from another TCA cycle as well. So complex 2 receives its hydrogen, protons, and electrons from FADH2. Now we see that complex 2 is not transmembrane spanning, so it cannot donate any or cannot pump any hydrogen ions into the membrane space. And so this is where cold Q comes into play. Cold Q, the electron carrier, has already taken the electrons from complex 1, and then it gets the electrons from complex 2 and transports them to complex 3. And in complex 3, the electrons are transferred and more hydrogen protons are pumped into that membrane space. This pumping of protons into that membrane space is creating a very positive gradient in the membrane space, and that's going to be the driving force for ATP synthesis. And so the electrons are all from complex 1 and 2 are now at complex 3. They're going to be donated to a cytochrome C, which is the electron carrier between complex 3 and 4, shown here. Cytochrome C will then donate electrons to complex 4 and hydrogen protons as well. Those electrons will combine to produce a water molecule. So oxygen associated with complex 4 is the final electron acceptor in the electron transport chain. So again, now we see that there are hydrogen ions being pumped in now by 3 complexes, 1, 3, and 4, creating a significant positive gradient in the space. And so actually, so the first 4 complexes are the actual electron transport chain. So now we're going to move to the 5th complex, and that is where chemiosmosis occurs. Chemiosmosis is the process where ions move from a high concentration to a low concentration, and they're going to do that at complex 5. That's going to be what drives the synthesis of ATP by ATP synthase. So what we have in this pathway, the chemiosmosis in complex 5 and the electron transport chain together are called oxidative phosphorylation. So it's called oxidative phosphorylation because coenzymes are being oxidized in the electron transport chain, and they're donating, when they donate electrons and protons, that's oxidation. And then during ATP synthesis, the ADP is being phosphorylated to produce ATP, so hence the name oxidative phosphorylation. Any questions about that? Does that make sense? Yes, no, maybe? Yes.
Can you, for complex number 4, what hydrogen is moving out? Like, where does the hydrogen come from that's moving out into the...
Moving into the space, it's coming from the... So remember that FADH can't donate hydrogen because FADH, it's not a transmembrane spanning protein, so it cannot push its hydrogen protons into the space. So there's going to be some hydrogen electrons floating around. So they're going to eventually go through complex 4 into that space. So what happens when the electrons are transferred from cytochrome C to complex 4, that is going to like transiently negatively charge the complex, causing a conformational change. So any floating protons are going to want to go through this channel here. Does that make sense?
Yeah, it makes sense. It's just I was just conceptually I can't. If
You don't know where, like, it's just like random.
Right, like if
random
we're using the
protons.
2 hydrogens to make the water, like that seems to me like you're using hydrogens. So like they wouldn't be they're being used. Like where's it coming from to move through?
Yeah, there's going to be more than just those, because of other reactions that are going on in the cell. So. Any other questions?
I have a question.
Yes.
So the number of hydrogens that are being donated, is that equal to the number of ATP that's being produced?
No. The number of hydrogens are different, and to be honest with you, I don't really know like the exact numbers. I've seen many different things. Like I've seen that there are 10 hydrogen protons being pumped from NAD and only 6 from FADH2, but I don't really know. I've never been taught or looked into detail in the number of actual specific hydrogen ions that are being pumped. All I have learned and always been taught is that they're being pumped however many, and it's creating a positive gradient in the space, and that's the driving force that's going to be used to produce ATP. Any other questions? And this, again, is just giving information about chemiosmosis. Chemiosmosis is that driving force because of the positive gradient of the hydrogen ions that have been pumped into the space. They now want to drive down both the opposite direction from high to low concentration to produce that ATP. So, in terms of the amount of ATP that is synthesized in this electron transport chain and oxidative phosphorylation, it has been scientifically determined that for every NADH, 2.5 ATP will be produced, and for every FADH, 1.5 will be produced. Now, these numbers were different when I was in school. They didn't know as much information as they do now. So, yeah. So, any questions about those numbers? All right. Check on learning. So, this is specifically talking about the carrier systems. What's the carrier between complexes 3 and 4? Cytochrome C, site C. What about the carrier of electrons from complexes 1 and 2? OQ, ubiquitin, absolutely. And what accepts 2 hydrogens and electrons from that? Cytochrome, which one you said?
Cytochrome C.
It's CoQ, I believe. Is it? CoQ. Let's go back. So here's FAD, cytochrome C is associated, so it's going to donate its electrons. Any questions? All right. What about these products? Tell me where these products are, what pathway they're coming from. Carbon dioxide. FADH2. NADH. Say again. Electron transport. I'm sorry, I said NADH. And water. Any questions about that? So, cellular respiration is the pathway from glycolysis through the electron transport chain. And so, this is just a little chart telling you or telling us how much ATP we get for each pathway and combined. From glycolysis, we know the final step of glycolysis produces an ATP and an NADH, but if we're starting from a molecule of glucose, of course that's going to be 2 ATP produced and 2.5 to 5 NADH. Can you guys tell me why it's 2.5 to 5 instead of just 5?
Determine whether it's aerobic or anaerobic conversion.
Absolutely. So if there is aerobic respiration, for both it's going to be 5. But because remember, if we run out of oxygen during exercise, that pyruvate that's formed is going to go to lactate, and lactate uses an NADH. Great, that was excellent. Any questions about that, guys? All right, so for pyruvate metabolism to acetyl-CoA, if it's aerobic respiration, which it will be, of course, going to produce 5 ATP, and then in the Krebs cycle, we get 8 GTP that's converted to ATP, 2 of those, and then 6 NAD and 2 FADH, which would equal 15 and 3 ATP molecules respectively. So the total potential ATP that is formed in cellular respiration from glycolysis through the electron transport chain is between 30 and 32 ATP molecules. Are there any questions? Yes.
For an exam, are we going to be expected to calculate the total ATP based on those given factors, or are you more interested in us knowing the actual pathways and processes? Based on
So,
what we're-
so I might say, how many ATPs are produced with 5 turns of the TC cycle. So you would take this information and be able to determine the number of ATP produced by the coenzymes. In terms of the pathways, just know the main features like the rate limiting enzymes and the main products in this case would be the reduced coenzymes. Any other questions? All right, I'll see you guys tomorrow. Have a great day.
That